So it's like
So the point I'm making, that's great, thank you. So the point I'm making is, in many situations, and the software will show you, all of these have been implemented. So while it's useful to have a rough idea of the theory, you don't have to be able to have the exact technical detail to implement one from scratch. So knowing about what these different techniques are and how they work is useful, but you don't have to have a comprehensive knowledge in order to use them. It's a bit like, as I say, if you drive a car, there's the skill in driving the car, but you don't have to build your car before you drive it. So it's knowing the techniques, but what I'm going to show you is ways to start using them as well, so that they can empower you to start using those things and start discovering new things. So does anybody have any questions? Has anything occurred to anybody overnight? Is there anything you want clarifying? And this is an opportunity for you to do that. I can go over things again, explain things again. Please ask questions.
I'm new in this AI thing. So if I have a data set and I'm not aware of much of its features, like I have a very limited set, but I want to extract some more new features which are unknown to me. So is it possible to...
This is causing an issue with this. No, I mean, we can just... Okay, yeah. So you want to find out, you've got a data set and you want to find out new features of importance about that data set. Yeah, so, but you have, it's a high dimensional data set where it has a large number of variables, you just don't know how they relate to the thing of importance. Yeah, so I can show you some more of that, and I will show some more of that coming up. But essentially the method we use is we look at each feature on its own in the first instance across the data set. We look at its ability to model an answer to a question. So say you have disease healthy and you've got all these variables. We test variable 1 against disease healthy, see how good it is at predicting it. We then test variable 2, see how good it is to predict it. Test variable 3. So in this way, we're testing out every single variable in the list, and we can rank them and see which are the best ones to use. In that way, we can start to see at a first pass what is important. What we can also do, though, is we can start building in a stepwise fashion models. So we could say, right, what's the best single variable? Okay, we find the best single variable, we keep that. We then add remaining variables to that to find the best pair of variables. So
like that, we are selecting variables which are significant according to our data. We can add it on in a model based on this.
Yeah, so we're adding things into the model to build the model up in a stepwise fashion based upon performance. So we start off with find the best single one, then the best pair, the best triplet, best quadruplet, and so on. And keep growing the model until we stop getting an improvement in performance. So that's another way of building up a model from these data sets. Some of the methods I showed you yesterday around weights and things like that, they have limitations for these big data sets, so I wouldn't use those. But that was just to illustrate historical stuff. Any other questions? No.
Could you please explain the activation function part?
Sure. So, activation function is a function which is the mathematical basis of the neural network. So this piece here is the activation function. So what it is, it's once we have the summed values, so we have this value multiplied by the weight, plus this value multiplied by the weight, plus this value multiplied by the weight, and then we sum that value. We'll get a value from that. And then we apply it to the activation function. So what that does is, okay, let's have a look at our value of the sum of the weights across here. Okay, so it's quite high. So just reading effectively off the graph. So the amount of activation is, if it's high, goes out as high. Okay. So the number that comes from this sum is passed on to this function. So if, for example, it's very low, then the output's low. If it's high, the output's high. So it's transforming that number onto an S-shaped curve. And that dictates what comes out of that node into the next layer. So if we look at these activation functions, here, if the value's low, the value's low, and it would just linearly relate. So that's a linear activation function. That has the potential to go to infinity and the potential to go to minus infinity. If you look at the sigmoid, it tends to 0, it tends to 1. So it's dampening down those high ends, which is good, because sometimes the model will run out of control. So if it's low, let's say it's -6, we get a value of 0. If it's -2, we get a value of about 0.1. If it's +4, we get a value of 0.99. If it's +6, we get a value of 1. So the number comes in, the activation function, that value goes out. Yeah? So where it starts to have an impact as well are with rectified linear units. So this is about a decision-making process. So here, let's say we have a value of -5 coming out of that hidden node. Nothing happens, we just get 0. And we get 0 at -4, 0 at -3, 0 at -2, 0 at -1, 0 at 0. And then only when it's positive does it start to go up. So that's immediately squashing that distribution to get rid of all the negatives. So what we're doing then is choosing these activation functions based upon how they modulate what's coming into them from the network. And then by using different functions, we can start to represent different characteristics of the patterns in the data. Yeah, there's nothing stopping us having multiple hidden layers as well. So we could have a hidden layer which is full of sigmoids, then have a hidden layer full of rows. That would have a particular effect on the network, but we could try that out if we wanted to see. There's no right or wrong answer. It's we don't normally say, oh, this is this sort of data, we're going to use 3 layers of a rello and 1 layer of a sigmoid. It's a trial and error approach as to which ones we use. What we tend to find though is 10H and sigmoid work better for biological systems because there's more of that noise. It's not such a crisp cut-off as in physics systems, which would be more useful for ReLU or in some cases linear. So it's just really how the numbers are transformed. Any other questions? I'm presuming we can start. I don't know if it's streaming. I'll carry on anyway. So the next method is an alternative machine learning method. It's in the public domain. It's been used quite a lot. And that's support vector machines. A support vector machine is a supervised approach, so we're telling it the ground truth. So is it this class or that class? We can generally only cope with 2 classes. And it doesn't make support vector machines will not make a continuous regression type analysis. So what it's trying to do is find that decision boundary. So if you remember yesterday I talked about decision boundaries. You have a cloud of data, the decision boundary is the thing that separates one group from the other through multidimensional space. So this technique is reliant on a kernel. A kernel is kind of a seed function. So it's a type of function class. So kernels are a weighted function which maps the decision boundary. So it's a bit like how I showed yesterday where the neural network maps the decision boundary by putting 3 sigmoids together. A kernel will use a function that does a similar sort of thing. So a support vector machine will use a kernel that does a similar sort of thing. And we can choose what type of function. So sometimes it might be a linear function, so that's where there's a straight line decision boundary between the groups. We can use a sigmoid, we can use a log, or we can use a radial basis function. So radial basis functions are quite interesting. They're contracting and expanding spheres. So what that's trying to do is creating a sphere, or a couple of spheres, which are trying to define the classes. And it's the radial basis, so the radial is effectively the width, or half the width of the circle, half the diameter of the circle. And what happens is that the SVM changes the weights of the function, effectively scaling the data through the multiple dimensions to maximize the distance between the points and the decision boundary. So it knows the distance from class A to the line, it knows the distance from class B to the line, and then it moves the line such that the distance between A and the line and B and the line is as big as possible. So what does that look like? Well, it's back to this just 2D plot of the population. What we have effectively is healthy, we have disease, and what we're trying to do is separate out one from the other with this line. So the first thing we do is we map the distance between each point in the population and the line. So we know the red lines have so much distance away from the line. These will be negatively weighted because they're on the wrong side of the line. And then we have the greens, which have so much distance from that. So we're mapping out that data space and the line's position relative. That effectively wasn't explained by the first rule. So that would look like this. So we take the X1 group, we split that into a pure cancer class, and we split it into a mixed class. Okay? And then we have our third rule, and now we're looking at the y-axis, and we're now saying, well, this is the feet of these people that we take as the point where they, in the space where they exist. So we're now saying if less than y1, then it's a cancer class, and if greater than y1, it's a healthy class. So with three nested rules, we've explained that whole population. So that's using a logical structure in the tree. Now broadly, if you look at this, you can see we have got effectively a decision boundary, which is sort of a little bit sigmoidal in its nature. So now we have a classifier. We can say, well, we put our cases through this decision tree and they get filtered out into particular groups. And what that looks like is something like this. So we take X1, our rule there, take X2, our rule there, take Y1, our rule there, and we have a way of classifying into the two types. The problem is, like everything else, this now is probably overfitted. There's a small population, it won't generalize well. So in the same way with a neural network, we have effectively mixed up the data into different groups. We can do this with a decision tree as well. So what we do is we take lots of trees, and what do we have when we have lots of trees? We have a forest. So a random forest is taking lots of splits of the data and then creating a tree for each split. So these all look the same in reality, they won't, but what we have in fact is multiple trees, each one has a different randomized view of the data. And by running it through that, we get a better, more generalized approach, more generalized model. So we don't get overfitting. So any questions on that so far? Okay, so there's a few other things we need to consider. And this is where we get into gardening and arboriculture. So, first of all, a tree can be very complicated. You could have, theoretically, as many branches in the tree as you have individuals. So this is a branch for this individual, this is a branch for this individual, and those branches get very complicated. So we can prune the tree to make it more generalized. We effectively take branches out and put them back into one amalgamated group, which may not be perfect, but it will do reasonably well. It will be more generalizable. So we can start taking out branches. We can effectively look at consensus across the random forest. We can merge trees into kind of one amalgamated tree, and we can have partial decisions on branches as well. I'm not going to go too much into that because it's a bit more complicated. Suffice it to say, random forests are a pretty good classifier. They work quite well. They can work well for relatively high dimensional data, and we've seen sort of similar performances in random forests to neural networks. It's an alternative tool. So that's the end of that lecture on the introduction to machine learning. I think we don't know if Adi needs to give his extra lecture. No, I don't know, maybe he's somewhere else. I'm just going to see what we've got next. I think it's... I can start. This is kind of quite contiguous. So machine learning for visualization, for undertaking analysis is quite a different undertaking, and it's probably best learnt by doing, and can take, those sort of coding courses take 6 months to run. If you want to seriously get into machine learning, ultimately using those methods allow you to automate processes. They will empower you to try out experiments. The methods I'm going to show you, certainly Weka is usable. People publish in calibration between those two populations. So normally you can only work on one or the other of those data sets. What batch correction does is effectively allows you to overcome that batch effect and normalize your points back so that they're all in the states. And then you can use something called combat to batch correct. That's a useful set of tools. Right, okay, let's try and get a new Google window. Well, there's nothing happening here.
I have internet connection to the modules.
That's taking you to a Udemy course. That's not what I want. That's why. There you go. So this is the Orange webpage, orangedataMining.com. If I go to This is just the page for it. It's about visual programming. Let's see. Right, so if I look at videos, there's a whole YouTube channel for this there we go. So we've got a welcome.
First you will see is a welcome screen. From here you can start a new date. You will see is a welcome screen. From here you can start a new date. Hello and welcome to Organised tutorial.
So this gives you tutorials, it tells you how to work through various methods, it tells you the basis of the methods.
Installed Orange. If not, installed Orange. If not,
and you can see it's got getting started with, it tells you about neural networks, it tells you about visualization, it tells you about neural networks, it tells you about visualization, it tells you about a whole range of different tools. I'll close that down. I've got Orange running here. Close that down. I've got Orange running here. Seems to be on go slow today. Here we go. So what I've got, the first thing is the splash page. I'll create a new workbook. I'm just going to skip that. So what you've got, for example, here is I've got a widget, and that says CSV file import. So that's pretty much the standard form of these sorts of data sets. So it's a comma, separated value, easily read in Excel, but also many text type formats like that with CSV as a standard. So what I can do is if I double click on that, I can select a, see if it's desktop. I can select a data file. I just want to Pops documents. Yeah, data for course. So I've got a data set which will show roughly how it works. What I've got is a set of PI3K, IgG, ER, and then P9. And then I've got a class, this is a classification problem. What else can I do? One thing which might be useful is a t-SNE plot. So if I click on help for this, it tells me all about the t-SNE plot. It's two-dimensional data projection. So it's t-distributed stochastic mode embedding. It tells me all about that, how I can do various things with it, and how I can pre-process this data, and so on. So it's a useful instruction how to use that, and that's a technique that's quite widely used. So I'm going to take the data into that. You can see it's running. Now if I click this, You can see I've got a two-dimensional plot of my cases. It has highlighted which ones are healthy, which ones are disease. This one's using three principal components. It's only 5 variables, 6 variables in the data, so it's not going to use many more than that. But I can increase that to see if it makes a difference. Let's take it up to 4. I've got distance metrics. I can exaggerate, and you can see by using more principal components it creates the shape a bit more. I can see potential outliers, I can see clusters are behaving the same way. Some things I can switch on and off, like the global structure, that hasn't made much difference there. I can choose to normalize the data or not. That can have a big impact. Is that completed? If it has, it's not very good. Probably does need normalizing then. And then what I can do to visualize this is I can set them as a different color, set them as a different shape, and I can show color regions. So there's not really much of a color region, although the blue areas you can see are shaded blue, and there's kind of an edge to our disease cases around here and a bit around here. So we can use it to get an understanding of what's happening in our data. If I'm using this for gene expression array data, what I usually do is pull housekeeping genes off the array, usually 15 housekeeping genes, and it allows me to run this on housekeeping genes, which means they should all be the same. They should be relatively invariant across a population. So if I get big differences in those, and I can see obvious groupings, then there's a clear batch effect going on. I don't want to do a t-SNE plot on every single transcript, but I do want to, say, pick a number of housekeeping genes to see if there's that structure in there. That's a useful tool. If I want to export this, I can put it to a note by moving the cursor over down here. I can also save the image as a graphic, PNG or JPEG or a PDF. So that's the first thing I can do. I'll close that down. What I can also do is I can use unsupervised approaches, so I can do clustering. So I can see whether there is a particular cluster in the data. So that's hierarchical clustering. I'm not going to go into that too much, but we can use it to do. And then I can. So the widgets for that are model. And you can see I've got decision trees, I've got random forests, I've got support vector machines, logistic regression, neural networks, Bayes. So I've got a whole set of different machine learning tools that I can use. I'll just maximize that. So what I'm going to do is I'm going to create a neural network. So what I need to do is take the data from my select columns, drag that into the neural network, and then double click on that. You can see I've got various functions. I've got a logistic, identity, TANH, relo. I can choose one of those. I'm going to take a logistic for now. So I'll take TANH for change. And then I've got a solver. These are kind of extensions or further developments around the entropy compared to the gradient descent method I showed before. I'm not going to go into those. You can look those up yourself. So and then I can choose the number of neurons I have in the hidden layer. So So if you remember, I said if it's a really big neural network, it can overfit. And in this, I've got 100 neurons in a single layer. If I typed into layer 100, 100, 100, I would have 3 hidden layers of 100 neurons. So that would be a really big network. So I'm just going to take that down to, I'll take it to 5. I can choose an amount of regularization. So regularization is a technique which stops the weights going too wildly away when it's training. It just prunes the network a little bit and prevents overfitting. So once I've got that, then what I'm going to do is test my neural network. And I take a line from the neural network, which is the predictions, into this widget. And I take a line from this, which is the ground truth. And what it does is I can now see that my neural network has various parameters. This is the area under a rock curve. So an area under a rock curve is basically the 1 sensitivity against specificity, if I remember rightly, so I'm just going off the top of my head. But basically, the area under that curve, the closer it is to 1, the better the classifier is. And anything with an AUC of 0.8 or more, it means it's explaining 80% of the variation in the population, regardless of the threshold which you choose. So this is not too bad, 0.766, but it probably could be better. Yep. So it's explaining about 77% of the cases. Now I can also, I'll do a random forest as well. And you know how I said about kind of using hyperparameter tuning? Well, I can start to build multiple models into this, and for my data I can try them all out and see which ones do well, which ones do less well, and cross-compare them side by side. So now, if you look, I've redone that, and my random forest has done less well than the neural network. I'm going to add in a support vector machine, and take that one to that one. So the SVM now has done much better. It's got an AUC of 7.795. So for this data, it appears that an SVM is a good solution. Now, once I've got that, I can do other things. So if I can do a rock analysis, so if you remember I said what a rock curve was. So it's basically sensitivity against 1 minus specificity, and the curve goes, should ideally go up to 1 and then across if it's a perfect classifier. But what this does is looks at different thresholds, decision thresholds. So the algorithm makes a prediction of a value between 0 and 1. And that's the probability that it's a disease, in this case a disease class. So if the value is 0.01, and it's a healthy class, that's very accurate, because that state is a 0, it's made a prediction of 0.01, it's just 10% error. If it makes a prediction of 0.01 and it's a disease case, then it's getting it badly wrong. There it's got 90% error. If it makes a prediction of 0.5, that's the decision threshold. So it's more, if it's over 0.5, it's more likely to be like disease than it is healthy. So by using that and changing that threshold, we create this ROC curve, or the system creates this ROC curve. And you can see this one doesn't give a key, which is a bit awkward. Oh, it does, yeah. So this one is the SVM, and that's doing much better across a lot of the curve, apart from the neural network does better in this bit. So we've got that analysis of our model, of our classifier. Do things like take out, let's go to data table. We can take out the predictions. So what we've got here, we've got our ground truth, and for this first case, the neural network predicted it as 1, which is correct, and the other two predicted it as 0. So the probability of a 1 here was 0.7, so it's got a higher probability of being a disease. The probability of a 1 for the random forest was 0.38, so you can see it's got it wrong, and the probability of the SVM of 1 is 0.41. So it's given me probabilities for each individual in the population, and it's got the variables as well. So I can slice into all my models and see how they work for each individual in the population. So something else we have in here, I think that's, it hasn't got so many. So if I click on options and add-ons, there's a whole list of extra packages which I can use here. And that gives us a lot more functionality, a lot more power, what we do. So, a few things that are important. So bioinformatics, that gives us access to a whole load of biomedical data sets, like from GIAN, so transcription, so expression array data and RNA-seq data. GIAN is a geographical package. Image analytics allows us to look at images and do thresholding and put filters in for images. Types allows us to build functional prototypes of models where we can build a model and put it out to a package. Spectroscopy, I'll open that one. This is specifically for analysing spectrograms, like mass spectrograms. So it's just taking a little while. These are not big things. Right, so if I restart Orange now, so if I go to spectrograms, oops, what's that one to? Spectroscopy, here we go. So I've got things like spectra, so I can load spectra in. It's a data table. I've got hyperspectra. I've got interpolate, so that allows me to do an interpolation of missing values. I can pre-process spectra by smoothing. I can integrate spectra. I can do peak fitting. I can create tile files. And I can do calculate signal-to-noise ratios. I can do partially squared analysis of the spectral profiles as well. So it does quite a lot. A line stack does alignment of spectral peaks as well. So we can align spectra a bit too. So that might be useful as well. What I would say is Orange is a good starting point, it's a good way to learn, but it doesn't have so much functionality. You're limited in the hyperparameter tuning. You're limited by how you can do that. You can't automate hyperparameter tuning, you're limited by a number of algorithms. Whereas Weka, which I'll show later, is a complete open source solution where people are adding new algorithms, new techniques all the time. So it's a kind of a community-based solution. Well, Orange is a community, but it doesn't have anywhere near as many features. So we'll cover that in the next lecture, and then we'll start looking through some other techniques as well. Any questions?
So like you have given the input of one CSV file.
Yeah.
So if you have like different CSV files for the same data, like different study with different parameters, so will it like get into same training set?
If you've got different parameter sets, then it's going to be difficult to merge those into one file which is contiguous, unless there's overlapping features. What I would tend to do with that is I'd run the two in parallel, and then look at what was found to be important in those two separate parts.
Then we can do it separately.
Yeah. You can load, with Orange, I think you can load some quite big files. With Weka, you can load big files as well. You may have to, there's some tweaks that you can use to increase the memory allocation of Java as well. But you can also tweak Weka to use GPUs as well. So, any other questions? Yeah. Yes, you can. Yeah, so there are so in Visualize, I think it is. Yeah, in Visualize you've got a heat map widget. So you can take, so if we take that value out of there, whether that will do anything. Yeah, it's not been scaled properly. So need to scale that one down a bit. Yeah, the values are not evenly distributed, but if they're on the same range, then you can select heat map for those variables. You can probably point it at particular variables and change the range for each one of those as well. But yeah. Any other questions? Somebody else put a hand, I think. Yeah. So it's normally set at 10.5 because if you have a continuous distribution between 0 being healthy and 1 being disease, it's half a distribution. Now, so let's say I make a prediction in first instance which was a value of 0.4 and 0. Yep. That's fine, correct. My second individual has a value of 0.48 and then there's a disease case. My third individual has a value of 0.6 and then a disease case, that's correct. What some people do is they move the threshold to improve accuracy either for positive or for negative cases. And reason they do that is, let's say you have a clinical study. You want to be more accurate at diagnosing people who have cancer and getting that right than diagnosing people who haven't had cancer. At the end of the day, if you've got somebody who hasn't got cancer and you get it wrong, so you have something the opposite has got cancer and you get it wrong, that's not a bad thing as somebody who you are saying is healthy and have cancer. So you change the threshold to give a better classifier in one particular direction. But yes, the 0.5 is the standard content.
So how if you have number of diseases, overpopulation, gender,
effectively put the algorithm scale. One of the things about machine learning is you don't have to have every sequence of continuous value, you can have it as a class as well. So you can have gender ending, let's say 1, 0. So you use that as a variable, put it into your model. So it's more flexible than conventional model. Okay, any other questions? I will say the best way to learn what orange might make things like that is to go and play. There's lots of data sets in the package which in the tutorials 1.22. So have a play with the data and try and things out. The guys that I actually do have done this course a number of times and they use it quite a lot. Even though it's a relatively simple solution, they do use it quite a bit. So. Okay. I think it's a good time to break for lunch. No. I think we have lunch. So just take a little bit of
as a application scientist for Thermo Fisher Scientific. And for 5 years, my job was to give trainings to troubleshoot. Right. So this was pretty much job was there in Thermo Fisher for different kinds of technology, whether it is imaging, whether it is mass spectrometry, right? So we had a technology known as high analysis, where you can do confocal imaging and simultaneously you can do image analysis, right? So I was supporting that. As I said, if you want to get some good money, you need to do some transition, right? So I did transition from application scientist to sales. So now I'm a sales manager in North, and I'm taking care of North region, including Nepal. And but it's my passion to always give having discussions about science with the students, with scientists, with teachers, right? So that's why we are here, and we are going to present again sample work for mass spectrometry, and we also like give you some insight about some technologies which is in the market, right? Which can make your life easy during experiments. Okay. So that's thank you.
Thank you, Dr. Anand, and it's a really great pleasure to have you here. And it's always a good feeling when sun was back. So welcome back to Allahabad University. And I hope we have more interactions. Okay, so I think we still have time for lunch.
Years. Last I came here in 2005 to collect my degree actually. I was very sober. I hope I will travel. I come back more. Still it's been warm here. There's a road going there on the science. It's all coming back. Between I've like you about so this is mass spec, right? So proteins you can get from any.
I think you can continue the discussion for 15 minutes. Lunch is on the way, so I don't want to hold you back another lecture. Might be hungry. People who are actually angry. Yeah, it's a fact. We are really angry when we are here. And this is a scientific fact. People don't get the road chances around. Free lunch chances per road, just in case you land at a JJ, ask your lawyer to send the parole application, just after lunch. Very high chances that they would accept that. Jokes apart, you can continue 15 minutes discuss. And you can also discuss, keep this formal, I mean, if you have doubts, Graham, all of you can sit down and discuss stuff. I'm a bit busy with that part. So I will be back after lunch. Thank you.
So most of you are PG students, MSc. So you all are aware of protein you can get from different kinds of sources. Bacteria, it could be I'm audible, right? Sorry. Okay. So protein, bacterial systems, and mammalian cells, right? And you are aware that for getting proteins from these different sources, the processes are different. Right. With plant, most of the people go for what? How to get the protein from plants? So if I want to extract protein, what I will do? Yes, grinding, right. Well, those are very hard cells, right? Mammalian cells, what you do? Just use some mild detergents and you get all the proteins. Here, again, those are very sturdy, right? What we do there? Any idea? It's not very much good and diametric, right? It takes lots of time, right? But the common application basically is sonication, right? They go for sonication because it's fast. But protein loss is too high, right? Recently, people have worked on some kind of GIANs, right? Those are some of the criteria where they dissolve cells of bacterial cells, right? And you can get the protein in say just 10 minutes. And the protein recovery is around 80-90%. As compared to sonication, where you get how much? 40%. Right? So these are the stuff. When you plan your experiments, you need to know about which is going to give you much better results, which technology, right? If you go with the little in the plant case, again the protein loss is too high, right? But for mammalian cells, having mild detergents, right? It's very good. It's the most advanced idea. So anyone for mammalian cells? Any PhD? Okay. So you are doing cell culture, you are doing.
It's in my last object.
Sorry.
It's in my last object.
Okay.
I'll be doing future.
Okay. So right now, what stage you are? So you are getting your cells, or
I'm actually completion.
okay. So now I am going to talk and all, and then you are going for the cell science and going to check, right? Okay. Great. So cell culture. Again. Mammalian cell culture is very different. You need to maintain certain kind of hygiene conditions, right? If you got the mycoplasma, you need to shut down your lab for 1 week. Fungal, 1 week, gone. Okay? Bacterial, not that much issue, right? Plants, they don't do much cell culture. Only up to certain extent they do what tissue culture. That's all. And those are very strict. Okay. So whenever you plan for your cell culture work, right? Plan it in such a way access of that lab should be strict. Okay. You should have a cell culture book at least cell culture book, and access to that also should be restricted. Okay. So those are the things which you need to take care whenever you are going to the expense. Right? Okay. Anything else? Any question? Anything which comes in your mind? Since we are mostly towards proteins, right? So do you guys know what kind of protein detection technologies are available? Come on. Mass spec is one. Right? Another one? Yes. Absolutely right. Anything? Microscope. ELISA. What you are doing? Cytosine. What they are right? Western blots. Right. So these are all the technologies we are going to see. Now, there is another technology which is in ELISA. Some of you are aware of it. In ELISA, at a time you can identify one single protein. Right, in a sample. Okay. But now the technology is so advanced. In a sample, you can identify up to 80 different kinds of tasks. What does technology is called? Luminance. Right? Imaging is now so advanced. Right now, this technology is used in diagnostics. Right? They are doing ISC, immunochemistry, and they are going up to 32 targets. in a single cell. So that the speed of diagnosing the disease is speed up, right? It's very high. Okay, they don't need to go for different samples. Need to take the sample again and from the patient. Right? Flow cytometry, you said right? Flow cytometry, people are targeting in a single cell 14 proteins, 16 proteins, 18 proteins at a time. Right? So these all are whatever technologies you are going to study or you are going to learn, right? Protein-related technologies are these. Okay. Now in a flow cytometer, now what they came up with, okay, they came up with not only you can identify protein, you can also identify specific genes through flow cytometry. What they did is they designed some primers, right? Tagged it inside the cells and then tagged that primer with some fluorescence signals. Okay, and now checking it with flow cytometry. So what is the advantage? You are studying a workflow, right? So you know in the workflow there are several critical steps, right? Some steps if getting affected, the next step will either get up in the upward direction or it will go in downward, right? So you can check the presence of that particular gene, whether it is getting more inside the cells, getting expressed, right, as compared to the control, and then you can check the downstream protein expression, right? So you can correlate. Okay. So these are the different kinds of technology. So my suggestion to you guys is that try to learn as much technology as you can. Okay. I will give you one example. When I was doing PhD, one of my friend, he was also doing PhD. Okay. Then he suddenly left. Okay. Then he suddenly left. So one day we met and I said, why you left? He said, I got a job in Thermo Fisher Scientific. Okay. I said, but you haven't done PhD. He said, no, I know mass spectrometry. And at that time, in 2006, he was getting 1 lakh rupees per month. That is too much. And his job was just to do the data analysis of mass spectrometry. And since he was so well versed in mass spectrometry that Thermo Fisher US, not India, Thermo Fisher US hired him. Right? So job opportunities are a lot. Okay? You need to have some technologies in your grasp. Flow cytometry, cell sorter, right? Great technologies. Mass spectrometry, great technologies. People are using now mass spectrometry in diagnosis, right? Flow cytometry, cell sorter is there. Imaging. Imaging is one very beautiful technology. If you are an image expert, you will get it, right? So I will say that please try to learn technologies as much as possible. Try to learn about a lot about them. They are nitty-gritties. So these are all software based, right? There are a lot of software jargons. There will be a lot of software analysis, right? There will be a lot of voltage, electricity, physics. If you go with the flow cytometer, you need to understand about the voltage, right? Right? So you need to know about physics also. Okay? So that is my suggestion to you guys, right? Okay, any question? It's only I am speaking, so any question from you guys? Come on, आप ही लोगों के बीच से हो भाई, पूछो। कोई क्वेश्चन नहीं? शर्माओ मत यार।
So why don't you brief about your PhD work?
Huh?
Why don't you brief about your PhD work?
Okay.
That you told
So my PhD work was basically mycobacterium. I was working on mycobacterium tuberculosis, and the work was to find out whether acetylcholinesterase are a better anti-tubercular drugs or not. And we tried also to find out the pathway, right? Which pathway they are inhibiting. Because initially my senior has found out that when she was working on Mycobacterium smegmatis, which is a non-pathogenic strain. So she found out that these acetylcoumarins, not normal coumarins, acetylcoumarins, these are chemical compounds, they are inhibiting mycobacterial growth. And smegmatis basically is a 10 times more resistant to anti-tubercular drugs which were available in market. From there, this idea came that since this is more resistant and it is getting inhibited in that range where the most effective drugs are working, so in tuberculosis, it should be more inhibition. And we found out, and we identified 20 compounds, effective compounds, out of 300 to 400 compounds. That these compounds are basically having more effect, more antibacterial effect, right, as compared to their other counterparts. And the role of acetyl group in the coumarin ring is important. Okay, so then we try to find out what is the pathway they are using. So one thing which we find out is that glutamine synthetase is also working as a acetyl group, acetyl transferase enzyme activity they are showing here, right? But till then, I finished my PhD, okay, because it took 5 years. Then my junior, she started working on that, and till she worked on till animal studies she did. There she found out that only 3 compounds are effective in animals, rest 17 are not working effectively, right? So that took another 5 years, 5 to 6 years. And then my guide got retired. So, when I joined Thermo Fisher, I found one technology known as high content analysis, right? Okay, so, and that technology was so good, so great. If I would have that technology, half of the work, the work we did in 10 years, we would have completed that in 5 years, right? So that was the advantage of that technology. And nobody was aware at that time in my institute, right, that there is one high content technology is available. Although there was one installation in CDRI of that machine, right, but nobody was that much aware. Okay, so that's why I always ask students to please learn technologies as much as you can. Okay.
What's the difference between the academic and the corporate world?
Look, in corporate, right? So how can I describe it? Right. So in corporate, what happened is you are responsible for several things. You need to finish some certain jobs in a certain time, right? So I have to do different kinds of activities in a year, and those are targets for me. I have to do that, right? Okay. And say suppose application specialist, application scientist, right? So in case of application scientist, he has to do certain kinds of trainings, some special trainings to some good institutions, right? So that is his target, okay? And those are time bound. So those time boundation is something which is you can find different in corporate, right? When you come to sales, I moved to sales actually, right? Because of better perks and better salary, right? So I moved to sales. In sales, I have targets, right? Targets of numbers, right? Targets of numbers, targets of marketing events, right? And then I have other targets as well, where I have to groom my team to become a next level manager so that they can be prepared for the next level manager, right? I have to develop some accounts as well, right? Accounts, for example, right now my initiative, our initiative is, you might have heard bio incubators, right? So my initiative is to collaborate with bio incubators, right? Make them understand about the technologies, right? Okay, so they can use more technology of my company in future, right? So education first, sales later, right? So yeah, sorry.
So I think sorry for breaking the flow, but sir is here for the entire day, I suppose.
Yeah.
Yeah, so you can keep interacting with him. Now I have come with the bad news and the good news. Bad news is, sir can't continue any further right now. Good news is,
Food is there.
food is here. So please join for lunch and be back by 2, because thereafter we have a presentation from, you can take 1 hour. So we have 1 hour. Right? Sounds good? Webcasting team, आप लोग भी आ जाइए. So be back by 2:15, 1 hour. I think it's sufficient.
Thermo Fisher Scientific again. Thermo Fisher Scientific not only produce products, right, sell products, they also try to impart their knowledge with only you need to do some registration. Presently, these courses are available. Antibodies, flow cytometry, protein biology, mass spec preparation workflow, right, and cell analysis. Okay, so these kind of please go register yourself and do the courses, right? Not only that, there are other courses also available, right? You just need to go to the application and techniques, and you can find a list of different kinds of lists, right? If courses are not there, there will be some information available for, right? About the principle, right? About how to, there are some tactics you might encounter there, right? Okay, how to do that particular. Okay, so that. I'm really sorry. This is a very old presentation, right? Okay, very old presentation. I thought I will give some different presentations, but I'm restricted to this. But this is a very old presentation. Okay, mass spectrometry sample prep workflow by me. Okay, this is one of my initial presentation, right? And one question you guys asked, right? The difference in corporate and government, right? After seeing this presentation, my manager said, what a shitty job you are doing, man. This is the presentation you are doing. After that, he arranged a training for me from Dale Carnegie, how to give presentations, right? So that is one advantage, right, in corporates where you are being asked to improve yourself continuously. We have our own university in Thermo Fisher University, where we can do different kind of courses. That could be how to write an email, how to do marketing, right? How to do a sales, how to do negotiations, how to do presentations, right? Okay. Apart from scientific trainings Thermo Fisher gives to their own people, we have a Gurukul session. Every week we are having a Gurukul sessions, right? And people like Dr. Arun, right, from all across India, they give trainings to one of the topics. To
sales people to marketing peoples, right? So not only science training, we also provide other soft trainings as soft skills training as well. Okay, so this is one of my early presentations. Okay, so sorry. So we all know, right? What is that? Cells, right? We are studying cells, whether it is cells, whether it is mammalian cells, right? It's all about cells, right? Life, okay? And what kind of studies we do? Whether it is genomics, whether it is proteomics, whether it is cellomics. Cellomics means study of cells as a whole, okay? So these kind of three different broader portfolio in life sciences you will find always, right? Genomics, proteomics, cellomics, okay? So this is some product information about Thermo Fisher, what products we have for different portfolio, okay? Now, so I think sir has already covered about what is mass spectrometry is, right? And I would like to keep it very interactive, right? Okay, I would like to keep it very interactive. So please question and answer, okay, in between, right? Okay. So mass spectrometry, we all know what is that. It's to study masses of molecules against charges, right? Mass by charge, okay? Biomolecules, not only biomolecules, in mass spectrometry, what kind of studies we do? I think you guys know that in oil industry, they do a lot of mass spectrometry, right? In dairy, they are doing mass spectrometry, okay, right? And yeah, chemical, and not only chemicals, even in pharma sector, when they are doing molecule analysis, they do mass spectrometry, right? Why mass spectrometry? Because you can, even if there is a difference of one diatom between two molecules, you can tell them apart, right? So that is the preciseness, that is the specificity, sensitivity of this technology. That's why it's very important. People are using in a diverse area of applications, right? Mass spectrometry. Okay. This is what I wanted to discuss with you. Biotechnology, pharmaceuticals, clinical, environmental, even geological, right? People are using mass spectrometry, okay? But here I am going to talk about proteins, right? Okay, a very different part of biotechnology or pharmaceutical, you can say. So, what kind of mass spectrometry we do generally? One is discovery analysis workflow, where you try to identify, okay, this is the target peptide, okay. Now, targeted where you already know, okay, this is my target and I want to see what, if it's happening, if it's expressing more or going down, what is happening to other proteins. So targeted analysis workflow, right? And in all steps, you will find common steps, right? Common steps of processing the sample, extracting the samples, right? All these steps are common for that. Only thing is, next, when you go to the machines, right, your objective is going to be different. They are what you are going to identify. Okay. So this is typical workflow for mass spectrometry, where you take some cells and tissues, right? You lyse it, right? Or you are taking some body fluid. I'm sorry, it's not very visible, even to me, even, right? So then you do some cleanup. Okay. Next step is some cleanup, right? Then separations of proteins, then purification, then you cut it down into peptides, right? And then you apply it for, then again after cutting it into the peptides, then you go for peptide cleanup as well, right? Some might have talked about C18 tapes and all, right? Right? Okay. So peptide cleanup, and then you apply it for mass spectrometry. So I'm going to talk about only about some kind of technologies which are available and which can make your life easy. Okay. So this is again cell lysis, right? Protein cleanup or protein enrichment, cleanup, protein detection. So this is some of the common steps which you do for any kind of applications, whether it is Western blot, okay, whether it is mass spectrometry, okay, you are going for different kind of analysis. So these are some of the common steps. You lyse the cells, you get out the proteins, right? If your protein is in a very small amount, you try to enrich the protein, right? So that you can get more proteins, right? Then you try to clean it up, okay? And then you go for the detection ki how much protein I have got, okay? And then you go for your end applications, whether it is Western blot, whether it is ELISA, whether it is mass spectrometry, okay, or flow cytometry, or whatever analysis you are going to do. So again, this is for mass spec specific, okay? So where you go for digestion, and then you go for the enrichment and cleanup, peptide enrichment and cleanup, and then you go for the analysis. So these are different kinds of instruments are available in mass spectrometry, okay? And Thermo Fisher is the market leader in this field, okay? So as I was discussing before the lunch, right? Sample preparation, right? So first step will be the sample preparation will be extraction of proteins, right? Taking out proteins from the cells, right? And the kind of cells, different kind of cells, different kind of way of extracting the proteins. Okay. So lysis method, it could be mechanical, it could be liquid homogenization, sonation, freeze thaw, manual grinding, right? That could be blander, homogenizer, sonicator, freeze dryer, mortar, and pestle grinding. Okay. Now, so it's always best to decide what kind of method you are going to use, what kind of method is available to you. Okay. Method also will depend the type of cells. You are going to do lot of plant cells, so definitely grinding and all is a suitable method for you, right? Now, and you can use a combination of physical and chemical methods, right? But recently, what has happened, as I was saying, that there are some lysis reagents some companies have discovered. This is the product which my company offer, but you will find the same kind of products in other companies as well, right? Okay, so I will just introduce you to some of them. One is beeper. Beeper means bacterial protein extraction reagent. Okay, if I say hyper, yes, insect. If I say peeper, yes, right? So purification, if you are doing lysis buffer for that. REPA and AMPER is basically for mammalian cells. Okay, peeper, tissue, right? Viper, yeast, right? So they have made, what they did is they studied the cell surface and different kind of cells, right? And REPA is basically is a formulation which is available on website, and you can make it on your own in your lab. Okay, companies also make that. Okay, but you can make it in your lab also. But all others are basically their proprietary one, which they don't share to what they are using. But mostly it's detergent. In some there are some enzymes are also involved. Okay, so bacterial, there are some enzymes involved. Okay, peeper, no enzyme, it's hard detergents. Okay, and hyper is basically a formulation of mammalian protein extraction reagent, some alternative formulation. Okay, so, and how does this going to help you, these kind of kits, right? What it does, as I was saying, that sonication, grinding, those mechanical methods, basically you lose a lot of proteins. Then you have to use a lot of samples. Okay, but these methods where you get some specialized formulation, okay, right, to extract the protein, gives you maximum recovery of your protein. So you have to use less samples for the protein extractions. Now, what if I want to extract in a cell, what? So suppose I want to extract mitochondria, what I will do? I want to study mitochondrial protein. Is there any method available? Yes, density gradient, right? Okay, density gradient is over there, one method where you can do mitochondrial protein extraction, right? And if you club it with some reagents which specifically put those cells, those mitochondria in a particular density, right, you get maximum recovery. Okay, so those kits are also available. Apart from that, there are some kits available. Say suppose I want to study membrane proteins, right? But how I am going to extract it? If I am going to use the detergent, just dissolve it. It will just dissolve it, right? I am not going to get it. Very cleverly, what they did is they biotinylated the amine of proteins. Okay, right? Okay, and then lyse the cells, and then they run it through nitroglidim, which is having affinity with the biotin, right? And then extracted the membrane proteins. Okay, so there are different methods of extracting proteins. Depends on what kind of experiment you are going to run, whether it is cytoplasmic protein, okay, whether it is mitochondrial protein, whether it is a Golgi protein, right? It's a membrane protein, it's a whole protein of bacteria, because bacteria do not have any organelles, right? So it depends on your experiment what kind of extraction method you have to apply. Okay, so these are some of the examples where the some with to ease out your workload. Okay. Yeah. So now I have extracted the all the proteins, right? All the proteins right now in open. What will happen? Any answer? What will happen to those proteins? They will get degraded, right? Why? Because there are several proteolytic and phospholytic enzymes which are also there inside the cells, but when you disrupt the cells, right, they get activated and they start killing your proteins, precious proteins. So now, just whenever you are going to do extraction of proteins, you should always include protease inhibitors, okay, in your systems. Okay, these are some of the inhibitors, and these are the drug targets which they target, right? That's phosphatases targets serine threonine phosphatases, right? Aminopeptidases, metalloproteases. So these are different kinds of inhibitors available in the market which will inhibit these proteolytic and phospholytic enzymes, right? And can prevent your precious proteins which you are extracting, right? Because cells, they have these enzymes available in their system so that they can degrade any protein, right? Inside the cells, that's a control mechanism, but once you open it, it's uncontrolled, okay? And they can kill any proteins. They can digest any proteins, right? So you need to protect your proteins, right? And there are combinations available with different companies of these inhibitors which you can use before extracting the proteins, right? These are some of the protease inhibitors. There are some combinations are also available, protease and phosphatases, right? Now I have isolated the protein, right? But suppose my protein is, I'm going directly for Western blot. I don't need to do any enrichment. I don't need to do anything here. I will just go run the gel, right? Will apply the target antibody that will bind to that specific protein, and I will detect it. Okay, my protein is present, right, in Western blot. Okay, but in mass spec, what happens is sometimes people try to enrich proteins. Okay, suppose I am working on mass phosphoproteins. Okay, phosphorylated proteins or phosphoproteins. If I am working, I will try to enrich my samples with those proteins only, so that other proteins can go out, and then my majority of my protein or the purified protein I should be having the phosphoproteins. Okay, so again, these are called prefractionation and enrichment, and there are different kinds of methods available: phosphoprotein enrichment, ubiquitin enrichment, glycoprotein enrichment, kinases based enrichment, active site based enrichment, and then there are some ion exchange methods. Okay. So our box, yeah, here it is. Okay. So where you do all these activities, right? Either you do all these activities in a column, right? Okay. So there are different kinds of columns available in the market. Gravity flow columns are there, and then centrifugal columns are there. These products comes in both the format. Okay. Either you can fill the resin in the columns, run over your samples, right? Phosphoproteins get trapped, right, and the rest will get out, and then you can elute your phosphoproteins, right? The same way there are centrifugal based purification of these of the enrichment of these proteins. Okay. Now, let me go. So there is, sorry, I was there. Depletion. There is one more method of enrichment, right? One more method. Basically, people apply for human serums. Okay. Sorry, I forgot that. That is called depletion method of enriching the protein. Where what I am doing is I am removing excess proteins, so I am depleting them, right? So that in turns I am enriching the low abundance proteins. Okay, so there are different kinds of kits available. So in a blood, basically it works for the blood. So what kind of proteins are there in blood which are abundantly present? Albumin, ma'am. Albumin, IgG, these are the major proteins which are there in the blood, right? So generally, whenever you go for the mass spectrometry, right, the sports, the drug test they do for the sports people, right? So they generally, whenever they go for the drug test, they take the patient's urine sample and blood samples, both. Right? And the Delhi Sports Injury Center, they have mass spectrometry and they try to identify from blood and from urine what molecules they are using, the sports people. So they use these kind of kits a lot to remove albumin and IgG from the blood samples. Right? That is one application. Okay? So, so that is one method. These are albumin and IgG, sorry, there are some product promotions there, here and there, right? I cannot get it rid of, okay? So, right? So these are the kits, not only in Thermo Fisher, but you can find it in some other companies as well, okay? So these are kits which can help you, again, to enrich your proteins so that you can do studies with less sample, right? Because samples are precious. Patient के ओके आपका blood दे दो अभी और वो काम नहीं हो पाएगा अभी और निकालना पड़ेगा, ठीक है? कितनी बार blood लोगे आप उसका? So that's why, right? So these enrichment kits are available. Okay, yeah, I was discussing about these, right? Okay. So this is one cell surface protein isolation kit. Biotinylation reagent to level amines on cell surface, and then you lyse it, interact with nucleoidin, get the proteins. Yeah, phosphoproteins enrichment kits, glycoproteins, ubiquitin, right? Now coming to sample cleanup. Yeah, right? So, sample cleanup, right? Why we need sample cleanup? Because down the line, we have used several different kinds of buffers. Okay, buffers has what? Salts, right? That might be not suitable when you are applying those samples to mass spec machines, right? So you need to remove, you need to clean up your sample, and during that time, it is also advisable to not lose your sample integrity, right? Okay, so these are the basically for the mass spectrometry, for ELISA, for Western blot, you may not need cleanup that much, right? Okay, but for mass spec, it's very crucial to have a very precise, a very good sample cleanup strategies. Okay, it's very required because then you will get noise in your data. You will get a lot of noise. Okay. Okay. So, as I said, there are different kinds of mass sample cleanup strategies are available. Spin column based, right? Small volume adaptive vessels, and plate based. You have multiple samples, right? So we call it high throughput. HT means high throughput. Okay. When I was discussing one technology, Luminex technology, right? So Luminex is what? It's a high throughput ELISA, right? It's a high throughput ELISA. In one, manually you are testing one target in one well at a time manually. But with that machine, you can do 80 targets in one sample, in one well, right? So it's what? It's a high throughput, right? So here also you can do cleanup in a high throughput fashions, right? Mostly here you can do 96 samples in one go, okay? So cleanup could be two types, right? One is desalting, another is dialysis. Can anyone explain to me what is desalting and dialysis? Sorry?
Dialysis you have
That's process, right? What they are, basically. That is process. I agree that you are putting a membrane and then there is a free flow of molecules. Right? That's process. But what they are actually. Dialysis is what, desalting is what. Desalting is straightforward. Removing salts. By what? By through resin. Right? Here you are using resin. Right? In desalting. Dialysis is what? Basically is buffer exchange and also removing big molecules. Desalting, you are removing small molecules, right? And dialysis, you are not only changing the salts from one salt to another salt, you are also removing big molecules as well, right? So there is always a cutoff of membrane, right? There is a cutoff. Okay, for 10 kDa, right? So 10 kDa will go there, less will be there, right? Okay, so that is what it is. So there are different kinds of cleanups available, right, with exceptional recovery. Even they can do 2 microliter sample. They can handle a 2 microliter sample, right, to 4 ml. Okay, so you have a precious sample. There are solutions there that you can still do desalting, right, even with the 2 microliter. So that is one thing. Sample cleanup again. This is, so dialysis, have you guys, anyone have done? Any person who, anyone? No? Up here? Oh, you have seen. So what you have seen? How people does? Yeah, there is a bag. They tie both the sides and then they put it into the buffer, right? Yeah, so that is called tube dialysis, right? That process is called tube dialysis. Okay, so in tube dialysis, you have generally it's called snake skin dialysis tubes. Okay, snake skin dialysis tubes. It's not snake skin, but it looks like that, so it's called snake skin dialysis tube. Okay, so they comes with a different molecular cutoff. Okay, and you put your sample inside that. Okay, tie both the side. Okay, it's an open-ended tube. Okay, you need to tie both the sides. Okay, and then you need to put it inside the buffer which you want to exchange. Right, that's a tedious process. Takes 24 hours. Right, 24 hours, sometimes 48 hours to change the buffer completely. Right, and to remove the unwanted biomolecules which you don't want to for the further studies. Right. Okay. Now. This is one dialysis apparatus, right? You put your sample here and put directly into buffer. It does in 6 hours. Okay? Why it does in 6 hours? Because it has a large surface area first, okay, right? And it can, and then you do it with the magnetic stirrer, right? They are also did, I also did, but due to large surface areas, right, the dialysis, the equilibrium attains very fast, right? So it does in 6 hours. Not only that, there are several others available. Here it is. Okay. So I need to open it. Here are the columns. Again, it's a dialysis. Right? You put your sample here, the rest of the buffer here, right? Put it there and just lay it. Okay? Simple. Right? So, these are the things. proteins, right? So cleanup right, you might get 200, 300 microgram protein in return, right? With these kits, basically, you can get 80% recovery with these, right? Okay. I will high hope with you, please. Yeah. But how, what does, which part of this concentrator does it? Sorry? There is a membrane actually. Yeah, there are membranes in these systems basically. Okay. So these are basically the examples of concentrator, right? You guys can check. Anyone? Yeah, please. You can run it. Okay. Yeah. Okay, no, you can run it to all the students. They can see. So you can see here that there is a membrane in between of the tube, right? So you put the buffer over there with your protein sample, right? Okay, and that membrane has a cutoff, right? So suppose that has a cutoff of 3K. What does it mean? It means that above 3K all will go there, the rest will be there. Right? So you can get your protein lower than 3K. Right? Okay? So what you are doing? Again, you are enriching your protein. This is also a method of enrichment, and this method is called concentrating the protein, concentrating the molecules. Okay. I will not go into that. Okay? So I have isolated my protein and I need to see whether my proteins are there or not. Right, so there are some of the methods which is known as silver staining and chromocy staining, right? So different kind of stainings are available in the market. You will encounter, even if you go for the SDS stage, a simple technology of protein electrophoresis, you will encounter these staining methods, right? So these are dyes which binds with the proteins, right? And then you can visualize it in a white light. Okay. Okay. So I will also not go here. You guys know about that. You guys know about protein digestion. Sir has explained it, right? Protein digestion, alkylation, guanidation. Right? These are simple chemical reactions, right? Which happens, right? And every company provides kits, provides right, for that, enzymes for that, right? You can find it in different companies. Thermo Fisher also provides these kits. So, yeah, so different proteases and protein digestion kits are available. There are two kinds of methods of tryptic digestion, right? One is in solution, one is in gel, right? So sir might have talked about it, right? Okay. So these kits, you can do it in your lab. You can buy these chemicals separately from different vendors, right? Or you can buy it from a company which can give it to you as a format, in a format of a kit. One thing, an advice, always try to buy a kit. Okay, the kit always have those ingredients together, and those are cheaper options, right? When you try to buy several chemicals separately, you spend more. Okay, so always try to buy kits, right? Okay. Now we talked about phosphoprotein. Now there are phosphopeptide enrichment kits available in the market as well. Okay. So these two kinds of kits, titanium dioxide and FENTA phosphopeptide enrichment. This binds with the monophosphorylated proteins, right? This binds with the multiple phosphorylated proteins. Okay. So again, now we did desalting in proteins. Now I want to do desalting in peptides. So for that we have C18 spin columns. So my assessor said that he had discussion of it with you guys, right? Okay, sure. One thing, two technologies now I would like to talk about with you guys. Okay, okay. Mass spec we understood, right? We are identifying proteins, right? Peptides in the format of peptides, right? I want to study, I have a two control set, right? One control, one test, right? Two tests are there, one control and one test sample. So how many times I have to do mass spec? Suppose in control, I have given some no any stimulants or nothing I have given there, but in test I have given some stimulants, right? Some reaction I have given there, right? And I need to see which protein is getting expressed and which protein is getting down-regulated, right? For example, so how many times I have to do the mass spec for these two samples? Definitely one, first time I will run control, then I will run test, right? But there is now a new technology available basically for mammalian cells, right? It's for mammalian cells. And that technology is known as SILAC, stable isotope labeling with amino acids in cell culture. Very simple principle, right? I am growing my control sample in normal amino acids, right? You know that when you are going to do cell culture, you need to grow cells, right? And for those growing cells, you need to provide them nutrients. And those nutrients includes amino acids too, right? So when I'm growing my control samples, I'm giving them amino acids. But if I am going to give test samples, right, I'm going to grow it to them with some isotopic, having some difference from lysine. Basically, we are using here lysine, right? So lysine का कोई isotope लेकर के I'm going to grow it to them. Okay, what will happen? One cell have lysine with the isotope, right? Test one. Another will be having lysine with the normal everything, right? Normal molecules. And now I can do what? I have extracted the protein. I have done all these steps separately, right? Mind you, separately I did that. All those steps, extraction, cleanup, peptide digestion, everything I did separately, and then I mixed it up in one sample, right? I incorporate it into the mass spec. What will happen? It will identify those isotopic amino acid differently from the control one, right? And in a single go, in a single experiment, I can do two samples at once, right? I can check in a single experiment, in a single analysis, I can analyze control versus test, right? So this is the technology, SILAC, stable isotope. This is one graphical representation. I have grown cells in light isotope containing media. Another one, isotope, heavy isotopes, right? Harvest, quantification, mixed cells, trypsin digestion, HPLC, and mix it up. And then this is how I get, okay? So suppose light one is having upregulation, right? Okay, and the heavy one having low regulation, low expression, right? So I can see a clear difference between light and heavy, right? So in a single experiment, here I has to run two different set of experiments, one with the control, one with the test, but here I have to do it once only, okay? So that is going to save my time, right? So that is one new technology SILAC has in the market. Now, I want to do, say, suppose 6 samples at once. What I will do? Yes, so these are tags. Basically, these are tags, ion signatures, right? Okay, I tag it. I tag the amino acids after doing the just a second, after doing extraction, denaturation, and everything. There, what I was doing, I was growing the cells. During that time, I was doing the tagging. But here, after doing every step of sample prep, right, I am treating peptides with different tags, reporters. These are called basically tag reporters, right? Reporter tags, right? So, and these all are having a difference of one delta only with each one, right? So these reporters gives you identity to a specific peptide in the mass spec. Okay. So from, say, suppose TMT16 has been tagged to the peptide of, say, P53, right? And the tag 2 has been tagged again to P53. It's not like they are discriminatory. They are not doing any discrimination in the peptide selections. They just go and they will tag to all the peptides. Okay, mass spec can read the sequence, right? So I have identified, okay, ये वाला जो peptide है ये P53 का है. Let's take TMT2 tag, right, of the same peptide, right, same peptide sequence, and let's compare, okay, their expression. ठीक है? तो मैं कितने sample कर सकता हूँ एक साथ? 6 samples. And now these kind of technologies has been so improved that you can do 16 samples, right, in a sample, in a one experiment, right? In one experiment, you can do 16 samples at a time. And think about the possibilities. मैंने TMT1 टैग वाले का एक प्रोटीन लिया और मुझे पता है कि वर्कफ्लो में दूसरा प्रोटीन है, राइट? मैंने उसका TMT3 का उसको उठाया, right, and I have compared the data. Okay, ये बढ़ा तो ये बढ़ा कि नहीं बढ़ा, या ये इसका expression करता है शायद, right? So I can check that, right? Okay, so possibilities are tremendous, right? Now the 16-plex assays are available. Okay, so up to 16 samples you can do in a single go, in a single experiment, right? Okay, so that's all from my side, basically. But if you have any question. Yeah.
This related method is here one sample and don't have strong statistical power. No?
One sample, so idea is that only that you need to grow first your cells. In Silac, you need to grow your cells first.
Yourself, I have one plate of control, one plate for I have one nitrogen normal and
yeah.
I will mix.
Yeah.
Statistically, I only one sample. But the statistical
So,
loss,
okay.
I don't have anything there. So that, if I say that sense, Select has not that much power to do logically measure.
No, it's okay. Okay, no, sir, look, these are not that much costly, first thing, right? To be suitable for your work. But it's, because what happens if I'm using 3 kDa, for example, and my sample 3 kDa is coming up, right? Because above 3 kDa, all has gone through it, right? So 3 kDa is there, I have isolated, concentrated it, I have taken it, right? Lower than 3 kDa. But what is there in the bottom? All kinds of proteins which is having high molecular weight than this 3 kDa, right? So you may need to choose then another membrane, right? Okay. Anything? Any questions? Okay. Thank you. So if you guys after MSc an advice, right? You want to join corporate, okay? After MSc, right? There are several companies who offers job, right? They do. You can do 2 to 3 months training also. Thermo Fisher also gives training, right? As a CSR activity, we call it corporate social responsibility, right? Okay. So under CSR things, we provide trainings, right? Okay, for 3 months, So that training includes flow cytometry, might be, and then sequencing. Most of the time it's next gen sequencing and real time PCR. Okay. Then after MSc, if you want to join any of the company, there are different kinds of opportunities available. If you are good in technologies, if you are really good in technologies, you can apply for FAS. FAS is Field Application Scientist, right? Where you provide technologies, information to customers, to scientists. You give trainings to them, right? Okay. And if they encounter any issue with that technology, right, you go there and you troubleshoot, right? Okay, so that is the job of FAS. Not only that, they also take parts in R&D work as well, right? That is the part job of giving trainings, troubleshooting and all. Main job is R&D. Okay, so that is one kind of job. But most of the time for FAS, they prefer PhDs. Okay, right, for FAS. Apart from that, after MSc, okay, so after PhD, you can apply for FAS job. Okay, after MSc also, if you are good, if you are extremely good, right, you can apply. Okay, now, after MSc, you want to make a transition to marketing and sales, like I did. I did after PhD and after becoming FAS, a field application scientist, right? It's better to have some trainings, right? It will be good if you have some marketing course, right? Other than that, there are different companies in the market who Thermo Fisher may not consider freshers, right? But there are different channels, different biotech companies, right, in Delhi, where you can join them and can have sales and marketing experience, right? They pay you, right? They give you daily expense, they give you monthly payout, right? But that has, as you know, in sales and marketing, you are having targets, right? You are having targets, so you need to do that. Okay, those targets you need to achieve. Okay, right? Then you can understand the market, you can gradually go up and can apply for any company, right? Okay, in my company there are several people who has done BTech biotech, who has done MSc biotech, right? After that they joined some companies, right? And after a few years they joined some other companies, then after a few years they joined some other, and then they come to Thermo Fisher Scientific, right? And right now they are in a very good position. Okay, so sales, marketing and technical. These three kinds of jobs are there available for science graduates and postgraduates. Okay, right? Just look into LinkedIn, right? Just try to, you might be encountering vendors, different channels who are coming to your lab, right? You can ask them, or you can ask those company people who are coming. Okay, I'm finishing, and Dr. Arun, he has finished his postdoc from BHU, and then he has a discussion with one of my colleagues. He said, yes, we have a job available. He applied and he got selected, right? Same case happened with me also. While I was doing postdoc, I had discussion with one of my colleagues, one of my service engineer, right? Okay, he came to my office to lab and was repairing -80. So I asked, your Thermo Fisher में कोई job है क्या? तो he said कि हाँ एक निकली तो है FAS की. मैं कहा मैं अप्लाई कर देता हूँ. मैंने अप्लाई किया, हो गया. ठीक है, right? So please make connections, right? Always try to make connections with people, right? That definitely going to help. Okay? Thank you. Thank you very much.
We'll move on to the next section. Yes. chances are that you won't get the certificate. So please try to, while I am going through this, please try to revise and ask yourself if you are clear with that.
So first, if I am to ask you, why omics? This is going back to the first lecture that Professor Sanjeeva gave. What's the point of studying omics? We are very happy, right? We were studying genes in isolation, proteins in isolation, and we have been doing this for a long time. Why do we need to study? Why at all go through all this headache? I mean, it's much easier to do a small test, maybe check for interleukin, or maybe check for anything. Why do we need omics?
So because eventually the whole pathway is being regulated by all proteins and genes and mRNA and microRNA, etc.
Right.
It's like a whole complex. If studying a singular entity will not define the whole biological aspect of how the modulatory role is going on.
Right. So that's what happened in the 1990s, and this is how this lecture would go. So initially there were departments of genetics, department of biochemistry. So those who are from biochemistry, forgive me, but this is what started off. In all universities across the world, zoology, botany, these are even older, ancient departments in science. Then there were genetics and biochemistry, and they started looking things at isolation. But if you look at it, any system does not work in isolation. A very simple example, suppose there was no railway in India. Would you be sitting here? Many of you would be sitting here, right? And you can tell that there are n number of reasons why. Because you will be able to travel here, right? Most of the thermal power plants run on coal that is brought via railway network to these thermal power plants, and these electricity fans and ACs, they won't work. So we are a network. We can say that we are all self-made and we are independent, but fact of the matter is, neither are we self-made nor are we independent. We are interdependent, and we are closely the way we behave, the way we move ahead, we are independent, we are linked to each other. And when you have to characterize multiple genes, when you have to study these impact, you teach, you have to learn from these multiple gene products or genes. So when we are using multi-omics or omics approaches, our primary area is usually human health, disease, agriculture. And why we need to study these multi-omics is because DNA, you see, is very static. You're born with a set of DNA, and throughout your lifetime, might be there will be some epigenetic changes, but overall your genome does not change much. But you do, right? We are born very tender, young, and gradually we age, we have gray hairs and wrinkled skin, and one day we would die. So to study DNA genomics, it tells us that, okay, this can happen. If you look at the point of view of a disease, you can say if you have this, like going back to yesterday's lecture, so Angelina Jolie had a BRCA mutation and she had a chance that she might have breast cancer. She went for mastectomy. She did not have. She might have had. But since it would be a triple negative breast cancer with very low poor prognosis, she went ahead to take a prior step. RNA, what it tells is what appears to be happening. RNA or transcriptomics, they never tell us what is happening. It's more like what appears to be happening. Because you see, even this enterprise, for what? MicroRNA. And what does microRNA do? It regulates other RNA and even sometimes proteins. So it's a more of a regulatory complex. So we know if you have an RNA or a transcript for a gene, it's very unlikely that whatever you see at the level of RNA, it will be seen at the phenome level. You might see the same you might see something extremely different. What primarily makes it happen is proteins and metabolites is more of a crime scene. When you look at metabolomics, you see it's more like a crime scene. The crime has already happened and you have metabolites all around. Then you start predicting that what has happened or sometimes what is in process, right? कहीं पे आग लगी है, धुआं चल रहा है, that is a metabolite, right? You don't know who set the fire, but you can tell definitely that it is on fire. So this is basically central dogma and how multiomics have evolved. And all of these work in conjunction with environmental factors. So you cannot forget. So it's part of our genome, the nature, and then you have the nurture. Together, they control. So proteomics was invented, the term was invented by Mark Wilkins in 1994 while he was still doing his PhD. So while those who are into PhD, don't feel disheartened. Much of you can do good things. So Mark Wilkins currently is a professor at working in Australia, and he discovered the term proteomics, and he was actually inspired by the word genomics. He thought, right? And people were talking of genomics in the early 90s. In 1994, proteomics burst into the fore. And I'll show you over a period of time how things have changed. So he defined proteomics as the entire complement of proteins including modifications produced by an organism or a system. What he perhaps missed out is at that moment or time. Because the beauty of proteomics, it keeps on changing. Right? When you wake up, you have a different proteome. When you go to sleep, your proteome is entirely different. So is your metabolome. Your genome and transcriptome, genome does not change, then it will be a very difficult thing for us, right? The guys in forensics will be banging their heads. Crime हो गया उसके बाद आदमी का genome change हो गया. You can't do that. So, but in proteome, it does change. It does change to even the smallest of stimuli. The proteome will change, and that gives us a very good way of understanding how events are unfolding in time and space. So I think I can go through these because we already know they serve as catalysts, they have signals, and we have gone through this. It's more of a repetition. So what I'm trying to tell you is how this field is evolving. So this is when the first term was invented in 1992-93, right? And you see there are almost 0 papers. And then you see how they have just shot through the roof. This is 2023, and in 2024, perhaps once we end 2024, we can get the data for that as well. So proteomics as science has gone up, and this is like almost like I think 20,000 articles per year that mentions proteomics as a keyword. It's not proteomics articles that have used proteomics as such. Proteomics as a keyword means proteomics is a big part of that work that they have done. And now we have HUPO, Human Proteome Organization, that is working. Professor Sanjeeva is part of HUPO. We also have in India, the Proteomics Society in India. All of you many of you might know. Those who do not know, do Google them up. And they are a good group working and spreading the knowledge of proteomics. And when we talk of proteins, the molecules that do work, you can easily say that if this is the protein abundance and this is the mRNA abundance, they are intimately tied to the cell state. mRNA measurements are often it's not a true proxy, so they are more of a proxy when you measure mRNA levels or transcriptome level for any disease. You do not actually get the true picture, you get the proxy, right? You know, all of you are from college, you know what is a proxy. What is a proxy? Proxy लगा देना मतलब क्या होता है? False attendance. So proxy is not, so proxy लगा देना is of course false attendance, but proxy is like you are trying to project that you are present in the class. So mRNA levels are almost always a proxy. Also the reason why I came into proteomics. My PhD had nothing of protein. Zero. My entire my PhD work was gene cloning and transcriptomics and real-time PCR and RNA in situ hybridization, and then I realized I'm chasing the wrong path. Then I did course correction, so till PhD I had zero proteomics knowledge. It's whatever I've learned after PhD. You're never late to take up the... So if you look at it, so this is how two cellular events are correlated, right? Protein abundances correlate. So this is a paper by comparative functional analysis of Prayagraj and Drosophila proteomes. If you see, for a specific pathway, if you look at it, the transcript correlation is 0.4, 0.47, with SAGE it's less, with Affymetrix it's a bit higher throughput, but we know a correlation of 0.47, is it good or bad? It's not good, not bad, I mean it doesn't really mean anything. But if I tell that between two species, C. elegans and Drosophila, when we look at protein, we find a strong correlation of 0.8. I have to take this call, sorry. Better? Okay. You can see a strong correlation. So this is for the entire proteome, and what they did was you can look up on this paper, this in class biology, it's a very old paper, yet I don't know how stronger correlation because techniques back then were not very strong. So there are two creams of proteomics. Can you tell me what is discovery proteomics? We have gone through so many lectures. Where's the mic? Yeah, anyone? Yeah, please. Can you pass the mic?
Hello?
Yeah. No. It's a good try, but discovery proteomics. So proteomics studies can be of two types.
Data dependent and data independent.
No, that's acquisition. You're right.
Trying to find out the unknown target in discovery proteomics. So we don't know about the protein which we are looking for.
Almost very close. So since we don't have much time, let me break the news to you. So discovery proteomics, where suppose you are trying to understand a novel pathway, right? You have two groups of cells. One is a cancer cell that is responding to a drug treatment. Another cell line, it's the same cancer, but it has become chemo-resistant. Now you want to understand why these cells have become chemo-resistant. A good way to do it is to study their proteome. You study both these proteomes. Cell line A, cell line cell line B. Same treatment, one is chemo-resistant, somebody is working on drug resistance, evolution of drug resistance in microbes. So suddenly you find the particular antibiotic resistance is developing, some antimicrobial resistance is developing. So you take both the proteomes and then you want to compare. So this is for exploratory investigation. So you know how explorers worked, how the British worked. They set sail with their ships, and whichever island they hit, they told this is ours, and they suddenly discovered things. So that is how discovery works. You discover. So until and unless they discovered India, India was not there, or so they believe. So So that's the joke between me and Professor Graham. So, so fact of the matter is, that is how discovery works. Something was already present, you did not know it was there, you used a technique, and you found out. Okay, this is what is happening. Then what is targeted? Here the question is, are proteins A, B, C in my sample? That means you want to probe that whether your friend went out with another friend last night, right? So you would try to ask other friends, देखे थे इन दोनों को? वही थे ना? अच्छा कॉफी पिए हैं? आइसक्रीम नहीं था, कोल्ड कॉफी थी. So now you are probing, now you are asking very targeted questions, right? So that's what you do. Suppose you want to, in a particular sample, you want to find out whether protein A is expressed. Now you don't want to know the differentially expressed protein. You just want to know how much of protein A is being expressed, how much of protein B is being expressed. So it's more like Western blot. But we will come to it why we would use mass spec and why not Western blots. Because Western blot, those who have a proper idea, I think many of you would have known what is a Western blot or an immunoblot. It is a very good way of telling that it is targeted proteomics, but only fact of the matter is, since it is not a high throughput method, is if you can do 100 Western blots in one day, that would be targeted proteomics. Whereas discovery proteomics is primarily studying the entire proteome, and ultimately it leads to tell things like differentially expressed proteome or binary proteins. And what are binary proteins? Binary proteins are proteins which are present in one group, absent in the other. Right? Binary, non-binary. Like nowadays, in the US, you have binaries and you have non-binaries. Binaries are like male, female. Non-binaries are gender fluid. They could be anything. Morning, female, afternoon, male, evening, female again. So, non-binary. I'm not kidding. You can just... But yes, you can have a laugh. So, these are primarily for hypothesis-driven investigation. So Professor Graham was telling yesterday that sometimes we do not need a priori hypothesis. When you are doing a discovery proteomics or exploratory investigation, suppose you want to study the impact of a drug on brain or impact of drug on a specific cancer or any system, and you want to explore that what all this drug could do, then it becomes exploratory proteomics. Now you want to understand impact of this drug on this particular pathway, where you will probe only for those specific pathway. Means your hypothesis is already set that this drug might alter pathway ABC. So my drug X works on pathway A, B or C. And this pathway ABC would have 1, 2, 3, 4, 5, 6 proteins. You use a mass spec to probe those 6 proteins. What you can do with this is, you can do relative and in fact absolute quantification. You can do absolute quantification or quantitation. You can absolutely measure the amount of protein each of these groups have. This shotgun proteomics, primarily nowadays the LFQ based proteomics, can give us relative quantitation, where we tell this is like fold change, 2-fold change, 3-fold change, but with respect to what? We do not know, right? I'm 2 times better than you or 3 times worse than you. That's the ratio. That does not tell us how much of the protein is present, right? When you do absolute quantification, you can tell 100 nanogram of protein X was present, or 1000 nanogram of this protein Y was present. And this is very important, especially when you're trying to develop a diagnostic assay, right? You have to do a LOOQ. What is LOOQ? Lower limit of quantification. So you have to make, when these kind of assays, the targeted assays, they have limits of quantification and stuff like that. So, and one more idea is why we need to, I think again I'm repeating what Professor Sanjeeva said, so there will be lots of repetitions as well. So if you look at genes, almost 22,000 genes, again new is 2020, 2,500, things are dynamic, but when you look at proteoforms, which are isoforms of proteins, right? Same protein, if it is phosphorylated at position 396, becomes a different protein, right? Suppose one protein has 7 phosphorylation sites. That one protein can, how many, now tell me what could be the possible, like in a simple example, suppose there is one protein that can be phosphorylated at 2 sites. So that protein can take up 3 proteoforms. 1, it's completely phosphorylated. 2, it is half phosphorylated. 3, fully phosphorylated, and within the half phosphorylations it can take up 2. So actually even with 2 isoforms, 2 phosphorylation sites, you can get 4 different types of proteoforms. That, and due to the structure function relationship, these 4 proteoforms can act in widely different ways, right? Cell division cycles, kinases. Look, if you go back to cell division cycle, we would see how phosphorylations can drive the cell cycle from G0 to G1, whether it will not divide. All these, especially important for cancer cells. So proteoform studies are helpful, and of course we have mRNA, we have SNPs, glycosylations, and proteoforms. So proteoforms can be studied using. So, when we design a proteomic study, science-wise, there are multiple things. I think these are, the most thorough table that proteomics can, what kind of proteomics exists. Genomics is pretty straight. Proteomics can be expression proteomics, where we do systematic proteomics or differential proteomics. When we do systematic proteomics, we just study a single proteome. Just imagine you just want to know what all proteins are being expressed in your particular cell line or animal tissue. You don't want to understand whether they are in disease state. You just want to study what is the normal. And sometimes we are so much hardwired to study differences that we sometimes forget that there are some things that are just normal. A reason why the longevity in their project is so cool, because they're studying how do we age. Very simple question, no mumbo jumbo here. What is normative aging? Forget aging and cancer, aging and gender. It's just how do we age, right? So there we will do systematic proteomics. We will do proteome profiling. We create a proteome profile that tells us that okay, this particular cell has 6,000 different proteins. End of story. Now then we can do differential proteomics. Here we can compare proteome profiles in different physiopathological conditions. Here you can bring in most of the proteomic study that you would find up if you look up on PubMed is differential proteomics. Impact of nerve gas, impact of cancer, impact of turmeric on disease XYZ, on brain, on liver, impact of COVID. That's what we did while we were studying COVID. So what happens in severe and non-severe COVID? So differential proteomics, we will compare the proteome profiles, maybe that of an organ, maybe that of anything. Like somebody was in the morning was talking of, bacterial cells, chlorella. So it can be from chlorella as well. We just want to understand the differences. Then this, and what does it tell? It can identify disease-specific biomarkers. if the proteins are extremely different, you see that protein X was not present in one condition, or it was faintly present, and suddenly it has gone up 25 times or 30 times, then immediately it becomes a biomarker. But only when you have a fair number of samples. It's not me, right? It has to be this, in a population, at least 95% people have to have that same protein going up, only then it becomes a biomarker. Next, protein, the structural proteomics, which of course is different from bottoms-up proteomics, but since you are into a proteomics lecture, you should understand that we use proteomics to study the structure. So people who design biosimilars, how many of you are aware of the term biosimilars? Yeah, what is biosimilar?
It is like a generic version of already marketed biological formulation.
Right. So you must have heard of tocilizumab and all these kind of antibodies that are used for therapies. Now, when you manufacture such a antibody, now antibody is a big molecule, right? And you need to characterize it. You need a proper structural study of it. So there are ways in, not of course not by bottoms up, but by a top-down approach. You can do structural proteomics as well. Then you can do functional proteomics, where you want to understand both by systematic profiling or via differential profiling how the subcellular proteins change. So you want to study the cell function. Now you would like to study the proteins in the nucleus, the proteins in the mitochondria, proteins in the cytosol, proteins on the cell membrane, right? Cell surface proteomics. That's also one way of doing it. Then you have metaproteomics. People who are trying to study, many of you would have heard the word metagenomics. So almost an equivalent term is metaproteomics. So metagenomics, there is an amplifying step. In metagenomics, there is no amplifying. That is good and bad at the same time. The reason is, in metagenomics, since there is lots of amplification done, sometimes you amplify noise. Here, since in metaproteomics you don't do amplification, the noise is cut off, but this is bad as well, because sometimes even if this signal is faint, you don't get any signal. I mean, if the number of proteins present for a particular microbe is less, you don't get any good amount of signal. So all these together can help you to understand protein-protein interaction, the mechanisms or functions of proteins, how proteins are moving around, compartmentalization of proteins, how proteins migrate. All these things can be studied. Also, an impact of microbes on gut ecosystem, the gut-brain axis. That is, you can all do that from the proteomic data. Sometimes you can also pick up genomic data and do something known as proteogenomics. A good example is, suppose you have anybody is suffering from cancer, right? What is a cancer? An aberration in the genome, and that aberration in the genome most likely has altered the proteome. That made the cell go crazy and divide like there is no end of day. It keeps on dividing. If we could understand for that particular cell what is the genome, then we will also be able to understand what is the proteome, right? That entire thing becomes proteogenomics. Very important when it comes to cancer. Apart from that, there are also terms like immunopeptidomics, where you look at the immune cells, the MSCs in particular. If you remember how MSCs function, you remember that there are small parts or small peptides that the MSC molecule holds on to, right? That are actually coming from the antigen presenting cells. Now, using immunopeptidomics, we can also understand what are those peptides that are being antigenic. So you can also understand how your body immune system is responding to sometimes even to cancers. That's why people do a lot of immunopeptidomics in cancer. That again can be translated to something known as CAR T cell therapy. Those who know it, else you can just look up on it. I think we have to take a small pause. You can have tea for 5 minutes and come back. The tea came 30 minutes late, so sorry for that. Broadcasting team, you can pause for 5 minutes if possible. Please have tea, and we'll be back in 5 minutes. and suggest where I am involved or Professor Graham is involved or perhaps Allahabad University. Till now, so far, we have almost like we're halfway through, like 2 days are over. In between, we have 1 day. A lot of things I'm repeating what Professor Sanjeeva had already said. The reason I'm repeating is because sometimes we learn more by repetitions. So, what does this slide show? Let's be interactive. We have 30 more minutes, so don't fret much. Whatever you do, I mean, I won't judge you. What does this slide show? I did not put anything per se. So, but what can you make out of it?
So one is the 2D gel-based proteomics, another is we can have the gel free. In that we have the mass spec and the microarray.
We don't have a microarray, but yeah, but kind of. So both are dependent on mass specs, but there are two ways of doing proteomics. One is gel-based proteomics, one is gel-free proteomics, right? Most of the but gel-based has its own benefits, like we were discussing in the early morning, for the first session in the morning, that how gels are a very cheap way to achieve things that even a mass spec cannot tell you. A mass spec can never tell you whether your proteins were intact or degraded. If your protein perhaps your and first dimension is isoelectric focusing. The second dimension is based on the mass, right? So first one is based on P isoelectric. The second one is based on the molecular mass. Now the problem with 2D was when you have to compare across specimens. Suppose you have 6 specimens of breast cancer and 6 peritumoral tissue that basically you take from just beside a tumor. Now if you have 6 different tissues, how many gels do you have to run? हाँ मैडम डिपार्टमेंट ऑफ ज्योग्राफी अनुपम पांडे अनुपम पांडे डिपार्टमेंट ऑफ ज्योग्राफी हाँ बताओ
सर दे चुके हैं अभी आ रहे हैं बहुत थक गए अंदर गाड़ी भी नहीं आने दी फिर तो दौड़ रहे हैं
ओहो चलो चलो थोड़ा वेट लॉस हो जाएगा कोई बात नहीं ठीक where was I? One more GIAN that I got in GIAN was if you are an admin, don't talk. Don't have your own talks scheduled. Get somebody else to do the talking. Anyway, so as a teacher you can't stop yourself from teaching. That's again. So if you have to have just 2 types of tissues, cancer, non-cancer. To achieve some degree of robustness in your data, how many gels do you have to run? gel and you get something, you would find a biomarker with just one gel? 2? No? So there is something known as biological replicate and technical replicate. Biological replicate is usually for these high definition data sets, usually set between 8 to 10. Means you have to take 8 to 10 individual samples. So 8 gallbladder cancer samples, 8 normal gallbladder. Right? Technical replicates, these kind of, so that is biological replicate. Technical replicate is triplicate, 3. So 8 multiplied by 3, 24. So if just for control you have to run 24 gels. Similarly for disease you have to run 24 gels. 48 gels. Now tell me what's the chance that all these 48 gels will be run under identical conditions, nothing going wrong from your side, from the electricity supply side, everything remains perfect. So there are lots of areas where things can go wrong, and things did go wrong. That's why this label-free approach was dropped. We took up a labeled approach that we call as differential in-gel electrophoresis. So we label 3 samples. You can see the red, green, there are 2, okay? So you have labels, Psi3, Psi5, these kinds of fluorescent labels are labeled. So when you label these samples, suppose the control sample was labeled with a blue bag, with blue fluorescence. The treated was labeled with red. And then you usually run another sample that is a mixture of both the two, and that we label as green. Now can you run the same gel on the same gel, because all these samples are now labeled with fluorescent tags? Just by scanning one gel at 3 different wavelengths, you can get results, right? So that way you are reducing the number of gels you have to run just by half. From 48 you drop it down to 24. So this is DIGE. You can look it up on DIGE, and after DIGE what you do, you pick spots. These spots are those spots which shows differential expression in protein. Of course we go for trypsin digestion, which I asked you not to use, but again this is a layman's term that most people use, tryptic digestion. And then you do ESI Q12 NS, which is ESI stands for, very good, electrospray ionization. And what is TOF? Time of flight. MS means mass spectrometry. So, and then you detect ions. Then by database searching, which basically MaxQuant will does, a software like MaxQuant will do for you, you can identify the proteins. Same thing that you can do in LC-based proteomics, in liquid chromatography-based proteomics. And in liquid chromatography-based proteomics, it can be either labeled or label-free. I think I was over here where Dr. Bagel, he was discussing about different kinds of labeling approaches. What kind of approaches did we discuss? So there are multiple types of labels. Maybe if you have time we will discuss. So there is TMT labeling. TMT stands for tandem mass tags. Then there is stable isotope labeling that we call as SILAC. So if you use it for animal culture, it's called SILAC, animal cell culture. If you are using for live mammals, you call it SILAM, stable isotope labeling for alive mammals. And what you do is you take isotope labeled amino acids and put it in the media so that when your cell starts growing, those isotopes are taken up. And then when you do the mass spec, you can identify just based on isotopes that what was my cell, suppose you have. So these are label-based technologies. Label-based technologies have issue, especially there is also ITRAQ, I-T-R-A-Q. So ITRAQ-based labeling, TMQ labeling, SILAC labeling, all these are labeling techniques. There is always an issue with labeling techniques. But labeling techniques are not completely reliable. They have around 70% efficiency and all proteins do not take up labels. Suppose you have used a specific amino acid. You won't label all the amino acids, right? You take 2 or 3 amino acids which you label in SILAC or SILAM. Depending on the presence of that amino acid in your protein sequence, your labeling efficiency will vary. Suppose you have taken valine or alanine or tryptophan and you have started labeling every tryptophan. You have taken the heavy tryptophan and your tryptophan wants to get incorporated into the cells. It will only be incorporated at specific where there is a codon for tryptophan. Suppose your protein has very few codons for tryptophan. Will it be labeled well? No. Second issue is, suppose it got labeled well, but when you go for the trypsin digestion, the peptides that are generated are either too big or too small. So you might end up with a big zero. That's why people slowly moved on to labeled methods, where unlabeled, label-free methods, where just by analyzing the chromatogram, you can, so much of the work that is being done these days is label-free. In label-free, all you need to do is, we'll discuss in detail today, we're running out of time. So in label-free, what you do is, we don't label the proteins as such. We take the proteins, we make peptides out of it, we inject the peptides, and then based on the chromatogram, we try to identify what this protein could be. So LC-MS tandem mass spectrometry coupled with an LC helps us doing this. So also we use the ESI Q-TOF-MS, sometimes we use Orbitraps. Then there is another method called CELD. Now CELD is actually something of a very old technique, but we don't use it anymore. So again, getting back to what is the basic composition of a mass spec. We have a sample, we have an ion source, you have a mass analyzer, you have a detector, and you have a data analysis. So mass spectrometry is basically this. Now, it could be a hybrid instrument, it could be a tribrid instrument, it could be any instrument. All depends whether you have one or multiple mass analyzers. So a hybrid instrument will have 2. A tribrid will have 3. An ion trap and Orbitrap together, that makes 2. Sometimes you also have a TOF thrown into it. The latest Astral is basically hybrid plus TOF. That gives you more sensitivity. So what does the ion source do? Mostly by electrospray ionization, which is a soft ionization technique, or by MOIRE, that is a matrix-assisted laser desorption ionization. It is a very hard technique, but in both techniques, you generate ions. You make charged peptides. You charge the peptides. And once the peptides are charged, via ion source, it will go to the mass analyzer, they will do a detection, they will analyze the mass, the detector will read the signals. Now I don't want to create a lot of fear in you, but there is a lot of physics and maths going around here. So usually we do, in an orbit trap, they do a Fourier transformation, right? It's called FTMS. That's the reason sometimes orbit traps will have FTMS, Fourier transform MS. We won't get into that. We will keep the biology side heavy. So we detect the different mass by charge ratios, and then the data analysis part, which we will take up tomorrow, which was supposed to take up today, we will take up tomorrow and decides. So again, if you basically divide mass spectrometry in today's time, there are discovery-based and hypothesis-driven workflows. Discovery-based workflows do not have any a priori hypotheses. We just want to understand how these things work, right? It is unbiased, and we do proteome or post-translational quantification. We just quantify qualitatively and quantitatively what is the proteome. Kitne proteins and how much of it. Sometimes we do relative quantification. Usually it's good for, and what we do is primarily the DDA and DIA. I guess Professor Sanjeeva also hinted upon it. So there are two ways by which an instrument can pick up signals to give you a very rough idea of how these things work. Suppose some ions are, you generate a lot of ions, right? In data-dependent acquisition, the larger the ion quantity, the more the number of ions, the more easy it will be detected. So that is data-dependent. Data-independent and in DIA-based methods, this is not a challenge. So in DIA methods, also you need DDA to create a library where the, I'm not going into the details, but thing is, DIA is dependent on DDA. First you have to run a DDA to generate a library, ion spectra, and then that ion spectra will be used for DIA-based methods. DIA methods, since we are not basing just by the number, the sheer volume, so these processes are very democratic, right? DDA is extremely democratic. More the numbers, more easily will be found. DIA is more like the Rajya Sabha, where you already have few selected members, you have a library, so that's how Rajya Sabha people are chosen, right? So we want to have a balance. So Rajya Sabha people come from the legislative assemblies, people are nominated by the president. So DIA is more like that. It gives you a better coverage because you can shut down the noise that is coming from some abundant proteins. Hypothesis-driven or targeted studies primarily work by two methods. One is the selected reaction monitoring or parallel reaction monitoring, SRM and PRM. And what we are seeing here is primarily three different types of instrument. One is primarily designed for DDA or discovery-based studies, where you see this is a triple quadruple. This is a mass analyzer, right? This is also a orbit trap mass analyzer. So these are all mass analyzers. This is either by DDA or DIA. Then you have a targeted type of studies. We can also use orbit trap for doing PRM, parallel reaction monitoring. For SRM, we use these kinds of triple quads. So there are these, one will select specific ions, then we will break the ions, and we'll come to it. So you can just remember for the sake of now that there are multiple methods. One is hypothesis driven, which is targeted, another is unbiased, discovery driven. And how does this end? This is very essential because we want to sequence, we do some shotgun sequencing of the protein based on the splitting of the protein peptides. So we do the second MS, the MS2. This creates the mass spectra. This mass spectra comes into the search engine like Andromeda, like tomorrow we'll discuss what is Andromeda and stuff like that. if you look at it, there are three levels of it. First is wet lab operation. Any lab who is perhaps passionate about the wet, you can do it in anywhere. You just need to have consumables. I think this box has a few, right? Next is the MS operation part. This can be outsourced to multiple facilities, like here in Uttar Pradesh, very close to us, there is BHU. Now, BHU has Sathi Center, Sophisticated Analytical and Technical Help Institute, Sathi. So BHU Sathi can help you with that. Also at IIT Bombay there is a Master D facility. At CDII maybe most of these facilities are central facilities because these are very pricey instruments running into crores, not lakhs. It's 2 crores, 3 crores, 12 crores. Then once they generate the MS spectra, this part again can be done in your own lab if you have a decent computer, a desktop with maybe 16 GB of RAM or 64 GB of RAM. Yeah, that's decent. 4 GB is not decent. So then you can run multiple spectrums, and nowadays there are much smarter softwares, MS Driver and stuff, which can run on much lower computing needs, and that can give you, so the better part, you can outsource. Top part you can do in your own lab, does not cost much money. I mean, it will cost in lakhs of course, but not in crores. And again, the computer operation, it's a one-time investment. If you can get the tools that Professor Graham is showing, with that you can sit at your home and do your own research. It's mostly DIY. So what can contemporary proteomics do is, even with the very cells and tissue, we can identify 2 to 3,000 proteins. I'm not even going to 10 stocks, which can go to 10,000. Even an average Q-exactive will identify almost 3K proteins. It will give you a very precise rate of quantification that is targeted proteomics. You can also identify different types of post-translational modifications. Very important if you want to study signaling pathways, right? Phosphoproteomics is a big thing. Many people working on cancer, just by looking at the proteome, won't get much of an answer. But the moment you look at the kinome, there are very fancy names to it. Like some people call it the kinome because this is driven by kinases. So if you look at the kinome, it can tell you what kind of proteins are getting activated, because multiple enzymes, if it is an enzymatic protein, it might depend on whether it is being phosphorylated or non-phosphorylated. Also, if you want to identify how proteins are interacting, you can do co-affinity precipitation, and then with those co-affinities, again can identify how many protein interactors do you have, right? So in co-affinity precipitation, this is a very old use of mass spec that I have been hearing about, because you don't need a very high throughput of this, but even with this, a good quality can give up actual physical interactors to your protein. So I guess we're almost on time. Just that we had one less lecture. But Professor Dr. Anils was an addition. So I have one more lecture pending, so tomorrow maybe morning we can start with that. 9, we can stop the request.
उस पे ओबीएस पे देखो ओबीएस चेक करो चल
So like we have 20 minutes, we can ask 40 questions.